Good morning. I want to thank the organizers for giving me the opportunity to share some of our latest results on elucidating the evolution history of Puccinigrammus formspecialis tritici. In this talk, I'm going to use PGT for the wheat stem rust fungus. We've been interested in the evolution of PGT for a number of years, and we've been working on defining different genetic groups within PGT. I've been interested in understanding this evolutionary process and how the different mechanisms to genetic evolution drive what we see. So a sexual mutation makes little steps, and those are represented in this phylogenetic tree on the right by variation at the end of branches. Sexual recombination and somatic hybridization are responsible for major changes in the tree, or the branch points, or the nodes. So we were interested in developing tools to better understand these recombination events, and especially if we could identify somatic hybridization events. It has been assumed that primarily sexual recombination was the driving factor for these branch points or new evolutionary lineages, but we really don't know how frequent somatic hybridization occurs. So this shows an illustration comparing sexual recombination and somatic hybridization. On the left, you have sexual recombination, and on the right, you have somatic hybridization. And the main point to take away from this slide is the difference in the outcomes. In sexual recombination, it's like shuffling a deck. So you pass on a haplotype, but it is changed. It's by recombination in the process. So it's not identical to either of the original haplotypes in the parents. In contrast, somatic hybridization, you pass on a whole intact nuclei. So the haplotype that you see in the parent is also the haplotype you see in the resulting progeny. So our approach was to use single copy conserved genes. So we mine genes conserved across basidiomycetes. We look for polymorphic between and within selected clades. And for this project, we selected 7 genes. We designed primers to conserved regions of these genes. And for the most part, we sequenced amplicons using Oxford nanopore technology. In the initial pilot project, we cloned amplicons, and we used Sanger sequencing. So this gives an overview of what the 7 genes that we selected are, gives locations both in supercontigs from the original PGT assembly, and chromosome that's been defined in the more recent Feng Li paper. Shows the amplicon size and the annotations for each of these genes. This slide shows the amount of allelic variation that we saw with a selected set of isolates, and we saw a range of 7 to 14 different alleles across these 7 genes. We also saw minor variance within alleles, which is shown on the right-hand side. This neighbor joining analysis, you can see allele A is divided into A1 and A2. So here's a summary of a representative isolate from a number of the genetic groups that have been defined over the last several years. And the point here is that each genetic group, each clade, subclade, has a unique combination of alleles across these 7 genes. So in the Feng Li paper, And as mentioned by Melania, they showed that UG99 lineage was a result of somatic hybridization between a genetic group containing the isolate PGT21 and some other unknown donor. And that haplotype A was passed from 21 lineage to UG99. But the origin of haplotype C was unknown. So this shows our results for the two haplotypes found in the 21 lineage. And the top is haplotype A and representative isolates, and sequence information available. Haplotype B, again, shows the allele for each of the genes, these 7 genes. In this next one, we show haplotype C and haplotype D. And the point here is that haplotype C probably came from genetic group defined as clade 2, in that we find the same alleles for these 7 genes in isolates of clade 2. And then corresponding, the other nuclei or another haplotype found in this genetic group is defined as haplotype D, as in the original figure. So, as a quick summary, we've shown that the evolution of UG99 lineage resulted from donation of haplotype A from lineage C and haplotype C from clade 2 or lineage A. So we were interested then in seeing if we could use this technology to identify other somatic events. So we looked at other genetic groups that we've been working with, and we found that 2 isolates, 13 Germany 16-3 and 13 Germany 16-4, which represent genetic groups 4C and 4E1, both share an exact identical haplotype. And then we defined haplotypes F and G as the corresponding haplotypes for the other nuclei of these 2 isolates. So in summary, we've developed tools to identify somatic hybridization events. We identified that lineage A, or genetic group clade 2, as the second nuclear donor for the UG99 lineage. Isolates of clades 4C and 4E1 share a common haplotype E, and were likely derived through somatic hybridization. This represents either a parent-child relationship or a siblings-like relationship where they share a common haplotype donor. I want to thank many people for this work, especially my group in the USDA Serial Disease Lab, Jerry, Kim, and Nick, Eugen, Pablo at the University of Minnesota, Batma at the University of Free State. And funding for this project has come from Bill and Melinda Gates Foundation, the Foreign Commonwealth and Development Office, and USDA. And also would like to thank BGRI for all that they have done over the many years. Thank you very much.